Well, welcome everybody. I am Keith Ezerick. I am with Cure Starts Now, also with the DIPG DMG Collaborative. We have the pleasure today to talk to Dr. David Zeigler. He is with the Children's Cancer Institute and the Children's Hospital of Sydney. And he's here to talk to us about some wonderful discoveries and some interesting research that he's been doing. You guys may be familiar with him, obviously, because he has done a lot of work in connection with some of the efforts of the DIPG Collaborative. He is also our 2021 Innovative Researcher of the Year. So with that in mind, Dr. David Zeigler, I'll let you take it from here and tell us a little bit about what you've been doing and some of the good research.
Thank you so much, Keith, and thanks for this opportunity to connect with everyone who's online. And thanks really for all the support over the years that's helped us to get to the point where we are now. With some really exciting developments, some exciting new treatments in the pipeline, and we wouldn't have been able to get where we are without the support of the DIPG Collaborative and the Cure Starts Now and all the people who've sort of believed in us, I guess, from day one. And I thought, as Keith said, I'll give a bit of an update. There's no slides, so this is just a chat. I'm happy, as Keith said, to take questions as we go. And really, I just thought it'd be good to both update you and give you a bit of a behind-the-scenes understanding of where we are and how we got here, and thinking back on where we started, which is, at a time where there was no research being done on DIPG, certainly in Australia at that time. We didn't even know if we could grow the cells from the autopsy samples that were being collected at the time, and we didn't have any mouse models. So we had to develop that all from scratch. And we did have some, key candidate drugs that we were excited about because, once we did get cells to grow, we started testing them, in the test tube in the lab. And then we finally got these mouse models up and running. And again, we weren't even sure when we started that whether these would reflect, whether we'd be able to use them, whether the tumours would grow fast enough, whether we'd get tumours, within a time that would be feasible to actually do testing in the lab. And what we actually found was that these models, when we established them, were really very much replicated what we saw in the clinic. They were very hard to treat. They grew rapidly. They developed the same symptoms that we saw in kids. And when you look at them under the microscope and you look at the genetics, they really were identical to what we saw in the clinic in patients. So they were a really good model. But what we also found is, as I mentioned, they were really hard to treat. So just as difficult as it is to treat kids with DIPG, it's just as hard to treat these mice with DIPG. And as a result, some of the things that we were most excited about, we quickly lost excitement with because the treatments failed. And again, just like we've seen with clinical trials, we were finding the same thing. And so while this was disappointing, it was actually not a bad thing because it meant that these were accurate models that, if they were too easy to treat, it would have not, reflected really what this disease is about, which is, such a nasty, difficult disease to treat. So we started looking at other, avenues, and the story that I'm talking about here, the polyamine pathway, this is a pathway that actually had been known about for decades as being really important for cells that are growing and dividing, and people had been interested about it in cancer. But, and drugs had been developed. So DFMO is an old drug that was developed in the 1970s, I think, and had been tried in brain tumours, but really didn't work, and people sort of lost interest in it. The only thing it's really been used for in the last few decades is for African sleeping sickness, so a very rare disease in Africa. And also there's a cream of DFMO used to remove excess facial hair, which is, kind of cute, but not very useful if you want to treat a brain tumour. So we actually had some colleagues here at the Children's Cancer Institute who there'd been a resurgence of interest in it in other childhood cancers. So another cancer called neuroblastoma. And there's some features that were shared between neuroblastoma and DIPG. A couple of times where you'd see a similar genetic sort of a pathway is activated. So, I had a new PhD student starting at the time. I thought, let's just give it a go. It's probably not going to work, but, we'll give it a go. Or at least she'll give it a go. And she was a fantastic, talented young girl who's still with us now, having completed her PhD. And what she actually discovered, and I should say, when you start any of these research programs, we really, we have lots of optimism, but we really don't know if anything's going to work, because to be at the cutting edge, you have to try things that no one's ever tried before. And what she really discovered were that DIPG, completely unexpectedly, are totally dependent on these polyamines. That if you even starve DIPG polyamines, they don't grow well. If you feed them polyamines, they grow much more rapidly. And essentially, the strategy that she developed was to use these two drugs at the same time. So one is a drug that stops polyamines being made by the cells, so stops what we call synthesis, and the other, and again, so that's the old drug DFMO, which we know never worked on its own, and her work explained why, and that's because you can stop the DIPG cells from making polyamines, but they basically bypass this by just pumping polyamines in from the microenvironment. And so she used this new drug called AMXT1501, which blocks the uptake, so blocks that pump and stops the polyamines coming into the cells. So that's the two strategies: one block synthesis, one block pump, and doing that in combination suddenly was really spectacularly effective. And as I said, these mouse models, which are extremely difficult to treat, we would get excited if we could see a drug just delaying progression of the tumour by even a few days. This was actually the most effective drug that we saw in these mouse models, where we were getting, mice surviving, and the tumour completely stopped growing. That extremely exciting results. And we've been working since then on trying to get this clinical trial up and running for kids with DIPG. And so just to update where things are at with that, and I know there's been a lot of sort of interest in this and support to develop this clinical trial, and a bit of confusion as well as to what's actually happening. So there's really two main trials that we need to talk about today. So one is there's the adult trial, which is for adults with any form of cancer, testing this new combination therapy, the AMXT1501 and the DFMO together. Because of our results and working closely with the company, what they agreed to do was actually extend that trial to include DIPG patients down to the age of 12 or in it. So a trial that had been open for adults 18 and above now has opened actually for children 12 years and above, or 12 to 18 with DIPG, or diffuse midline gliomas. So that's very exciting. And there has been some issues and discoveries that have been made through this trial, because this is the first time this therapy has been used in anyone anywhere in the world. And what was found is that, the first, with this sort of trial, you've got to start at a low dose level and gradually build up. And what they found was that as they built up the levels of the two drugs, both given orally, they started to see some toxicity, and that toxicity was gastrointestinal. So it wasn't tolerated to be able to take them both orally. Now, the simple thing to do at that point would be to say, well, we can't go up to a very high dose with both drugs, so we'll just drop the dose to one that's well tolerated and we'll leave it at that. And that would have been actually what most companies would have done and moved forward. But I think, and we worked with them on this, they took a very forward-thinking approach, was to say, actually, we could do that, but we actually don't think we're at a high enough dose level. And if we want to treat tumours like DIPG, we're going to need higher dose levels. And, that was partly of the work we've done, showing, exactly what the right dose is, what the right level is you need to be active in these tumours. And so to address that, what they've done is develop a new formulation of the DFMO. So the AMXT1501, that drug's still given orally, but then the idea is actually to give the second drug intravenously. It's a little bit complicated because it's not just, something you come in for once a week or once every few weeks through a drip. It has to be given continuously with a pump. So a pump that you basically carry around with you, and this drug gets infused continuously, sort of 24/7 for 4 weeks at a time. And so there's been these things take time, and there's been a lot of time that was taken to get approval and get that change in the formulation and get that those changes to the trial made. And I'm pleased to say that's now been done. And the first patients have now actually been treated with that intravenous formulation of the DFMO together with the oral formulation. And so that is now continuing as that adult trial, including patients down to 12 with DIPG. And so we will now start to understand whether this is more effective and less toxic than giving both drugs orally at the same time. And the other good news about that is that trial it was open just at MD Anderson, but that's now opening at 4 other sites in the US and also here at Sydney Children's Hospital in Australia. So it's opening. I should say I'm not directly involved in that trial, so I just understand what's going on, but I'm not actually running that. But I'm opening that at Sydney Children's Hospital. It's also opening at Cincinnati, Mayo Clinic, at the University of Washington, and there's one more, Fred Hutch, as well. And so, as in those places, as I said, it'll be open for any adults with any type of cancer, as well as kids down to 12 with diffuse midline gliomas. There's also the potential for compassionate access, which the company is looking at for younger kids, and I understand they're looking at actually treating the first kid with that on a compassionate access basis, and that their plan is, again, from what I understand, is that will be at those institutes that are actually opening the trial. If there are younger kids who aren't eligible for the trial, they will look at the possibility of compassionate access.
Well, that's certainly good news, and it certainly gives us some encouragement from a study that I mean, my goodness, when was this started? I think this was what, 2017 or 2015, I think, when this process started.
Yeah, it's been several years, and it's been at least a year of just that change in the formulation and moving from that oral to the intravenous, and so it is a slow process, but I think particularly our results in the DIPG mean that should help speed things up, because we have a specific disease where we're really seeing specific activity, which makes it easier to get patients recruited onto this sort of trial.
Now, when you were looking at the results with the mice, was it 2/3 of the mice that you saw this increased survival time with it? And did you see any type of recurrence from it at all?
Yeah, so at that time, we have a sort of ethics approval of how long we can treat the mice for. And so basically at that end of that time period, or at the end of the observation period, even after we stopped treatment, we did actually, as you said, see 2/3 of those mice had survived at that time point, which, as I said, is really unprecedented, at least in our hands, for any of the drugs that we had tested previously. That even, we would usually see in a really effective drug that we might be able to extend survival, but ultimately, the tumor would progress. So these mice all still had tumor. So it wasn't as though we were completely eliminating it, but we were completely stopping it from growing and stopping any symptoms from it in those numbers of mice.
Well, and I think that's, I mean, just in my short time in this of 16, 17 years of watching that, I don't think even I've seen that or heard of that. What I saw just an increased survival by a couple of months or something like that. So that's kind of interesting as well.
Absolutely, yeah.
You obviously talked a lot about DIPG, but we also get the question of is this applicable to DMG, and to what extent that this can extend to other types of brain tumors? Obviously, you alluded that they're looking at this trial for many other cancers as well, so it's got really those long range impacts to it. But is DMG a reasonable conclusion to this too?
Yeah, so absolutely. So I should say, so there's that trial, the adult trial, which is opening down to 12, and then once we have that data and we've established, and we need at least 6 patients to be treated with this intravenous formulation. Once we know that, then we're actually going to move forward and open through the Connect Consortium a larger trial specifically for kids, so for all ages, with DIPG, and when I say DIPG, I mean all diffuse midline gliomas, anyone who fits in that category. And yes, absolutely, we are, as I said, really excited about these results and have started looking at other sorts of brain tumours because, and I know, Keith, you are a strong advocate for this. If we find any treatment that's effective for DIPG, then this is really surely going to mean, if you can treat the most difficult to treat cancer, this is going to have implications for other cancers as well. And we've started looking at other brain tumours and have seen some really exciting results there. It's a little bit different to DIPG, so sometimes not quite as effective, but still seeing some similar sort of range of efficacy, and also potential for different combination therapies. There's a different standard of care, for example, for medulloblastoma. So we're looking at whether standard treatment for medulloblastoma like chemotherapy can actually make this work in that disease setting. So yes, we're very excited about this for other tumor types as well.
I have a couple other questions. One of the questions that we're seeing is this needed to continue? Do you have to keep the dosage going, or does the tumor start to grow back? I mean, is this basically going on forever? I mean, how does that stop that, and what have you seen on that?
So yeah, really great question. So in the mice, it's always a little bit different to what you do in kids. So we in the mice, we generally have treated for a long time. The PhD student, Amina Khan, who I mentioned, unfortunately, because this was a daily dose, she basically had to come in every weekend for months because all the mice were surviving. So she didn't have much of a break at all. In the kids, the plan is to continue as long as it's working. So we wouldn't necessarily stop at any point, and we would, yes, continue as long as it's working. The standard for any of these sorts of trials using a targeted therapy or a therapy that's relatively non-toxic is you treat, most people are comfortable to treat for up to 2 years. And then there's always a decision point at that time. Do you continue after that? I would say if you have a patient with DIPG who you have treated successfully for 2 years and it's working, at that point you would want to keep going. But hopefully this would be a great problem to have if we saw this treatment was working so well that we're thinking about, can we stop it? That would be a really, as I said, a great problem to have.
Well, and you mentioned that they went to this continuous pump option to effectively keep that dosage high. You also indicated that wasn't a normal conclusion. It was actually something that you felt was positive and kind of a partnership coming from the pharmaceutical company. Do you think that there'll be eventually other ways of delivering this other than IV? Is it always going to be kind of that continuous pump? And then second to that is do you think that there may be some discussions as to taking each one of these agents, whether it's DFMO or AMXT, and maybe putting it with another combination? I know that I've seen a trial already coming out that's using DFMO as a separate thing. What do you think of the possibilities there?
Yeah, so I think at this point, I think it's, if the pump works and that's the way forward, then I think that's the way that it will continue for the foreseeable future. I can't think of any other way, to address this issue to get the high enough dose. But yeah, absolutely, we are looking for the best combinations and the way to make it work even better. As I said, we had 2/3 of mice alive, but they still had tumour. So we're actually now looking in the lab at least, and we have some data that can show that actually if we can find not just combinations, so drugs to be given together, but actually you give one set of drugs followed by another set of drugs, then that may actually make it work even better. So absolutely, I think that will be the way forward. It's not for the clinic yet, so we've got to take one step at a time. But we are working hard in the lab so that we will have those discoveries when the time is right in the clinic. Then that would be what we would see as the next trial.
Okay. With that also, is there any discussions on that with methionine type of setup with the AMXT and the DFO combination?
Yeah, so that's one of the things that we've started to look at, because it's a very similar pathway, and there's been some really interesting results looking at the effect of methionine on DIPG growth, and reducing the levels of methionine can inhibit DIPG. So that's actually, you're exactly right. That's one of the combinations that we are starting to look at.
Okay. I know obviously you talked about several other types of research projects that's worked on, and a couple other compounds that may have some interesting options for treatment. Can you talk a little bit about some of that?
Sure. Yeah, so we have so I guess there's 3 other trials that are just worth mentioning based on some of the discoveries that we've made, and which people might have heard of. So there's a drug called ACT-001, and again, when we tested these cells in the lab, started screening drugs, many years ago now, we found this natural compound parthenolide to be one of the most active drugs. But again, parthenolide, we couldn't really get at high enough levels to get into the brain and be active in our mouse models. But ACT-001 is a new drug that's basically been developed as a synthetic form of parthenolide that is much better solubility and can get into the brain much better. And so we have worked with the company who makes that to do this trial, which also has taken a long time. The drug is manufactured in Wuhan, so we were hit by COVID, but we started at a very low dose level because we got into kids very early, while the adult trial was actually still going. But as we got data from the adults, and as we've increased the dose in kids, we've actually started to see some efficacy. So, again, which is almost unprecedented in the clinic. We've seen at least a couple of cases where we've seen the tumour respond in kids. So this is really exciting. This trial has been open in Australia only, but because of these results, we're now working with the company and again with Connect to have this as one of the arms of a bigger trial that will open hopefully around the world with this year through that consortium testing this compound. The other
is that with the target study? Is that
with the target study? So because this compound doesn't have a direct target, so it's not a direct gene that we're targeting. So this will be through the target study. It'll actually be for those tumors where we haven't found a specific target. So the target study, for those who aren't aware, will actually look at the genetics of each tumor and screen patients to arrive to the therapy that matches their genetics. So for those kids where there isn't a specific genetic match, we're looking at ACT-001 as the drug that they could get through that because it's not targeting a specific gene. And then there's another drug, CBL0137, again, that came out of the drug screening that we've done, where we're working through the COG, Children's Oncology Group. This is what we call first in child, so it's the first time this drug has been used in children. There's been a long pause on that drug, so we treated the first 9 patients, but we think we found the right dose level, but that has taken a lot of time for them to get regulatory approval and sort of ethics approval to move to the next stage. But hopefully that will reopen again in the next couple of months, and we'll be able to start retreating children again. There will be a specific group focused just again on DIPG and diffuse midline gliomas for that drug, which we found to be particularly active in that disease. And actually, it's the K27M mutation that seems to be the driver that means that kids are more likely the tumors are more likely to respond to this drug. If you have that mutation, which we know is the driver in DIPG, then that makes CBL0137 more active. So that's another trial, which hopefully will become available again in the next couple of months as soon as we get the regulatory approval.
And you mentioned these mutations, if I may. When you went through the mouse models, in each one of these types of studies, did you look at many different types of mutations? Even including the H3K27 and so forth, so on, or was this were these models different for each one of the trials? How varied was that?
Yeah, so some of the modeling we do in the mice, and we're restricted with the number of different models we have mice. When we're talking about DIPG and diffuse midline gliomas, we've only got a few sort of really good models that we can test. A lot of the testing is then done in vitro or in the test tube, where, for example, we can take cells and we actually remove the K27M mutation, that key driver in DIPG, and we find when we do that, they're actually the CBL0137 doesn't work as well. When you switch that gene back on, then the CBL0137 becomes more active. So it's exactly the same cells with or without the mutation. We see a difference in response. So that's really one of the clues. There's several other tests that we did, but that's one of the key clues that showed us that this drug actually is more active in tumors like that have this key genetic change that we see in DIPG.
And then with these mouse models, is this being used from, obviously you're growing the tissue there. Has it also been tested in, other formats, whether it's a biopsy or, that type of tissue, or is this just?
So most of the models we use are actually taking tissue either from a biopsy or from an autopsy specimen. And so it's actually parents, who helped us set up that program to get these specimens, whenever we could, and we're incredibly grateful for that. That's allowed us to generate these models. So that's the mainstay of the models we use. We've also, and now again, thanks to collaborators overseas, building what we call immunocompetent models. So models with a normal immune system, where we've actually got the tumor that's been, so basically mice that have been engineered genetically to grow tumors. So when we use an autopsy or a biopsy specimen from a patient, we can only use that in mice that don't have a normal immune system, because if they have an immune system, they reject a human tissue. Whereas these newer models are developed in mice with normal immune systems, and that then allows us to test drugs that might work via activation of the immune system. So it allows us a whole different way of trying to come up with other treatments, which is a good segue into the third or the fourth trial that I wanted to mention, which is CAR T cells, which we've just opened here in Sydney, which is very exciting. It's really based on Michelle Monge's great work and her team there, showing that these GD2 targeted CAR T cells really were spectacularly effective in her mouse models. And we've tested and developed this trial using a similar, slightly different, but similar type of CAR T cells based on that research. And that will now allow children in Australia to also use this, to have access to this new therapy as well.
And that is only for within Australia, correct?
That's right. So unfortunately, at this point we can't take patients from overseas. But again, I think the ACT story is a great example of where we've started with the trial in Australia, and having seen some results, we're then able to expand that overseas. So, I'm always reminded, when I get emails from parents, from around the world about the urgency, not just that I see here with my patients, but that this is really a global problem. And so we always think globally, but sometimes we do need to start locally. But it's about finding the best treatments and trying to expand that as rapidly as we can.
Well, we appreciate you guys doing fantastic work. I have one final question that somebody posed. It's going to be the hardest question that you have for the entire time. Which one are you more excited about of everything that you've said here today?
Yeah, that's a great question, and that's one I have been asked, and one that I asked myself. And I've said, actually, it's a little bit like asking me to choose which is my favorite child. Um, so, I am excited about all these treatments. And, in terms of, as a parent or, trying to choose between one or another, I think, ultimately, if any of these work, if we knew that they worked, they would be out there already as a standard treatment. So we always have to bear in mind that they're all experimental. We need to test them, we need to try them, we need to get as many children access to them as possible, but we always have to remember that they are experimental. So if we don't know that they would work, while we'll have great hope in each of them, we don't know until the trials have been done. And I think that's another really important message, is we really need to get treatment done on these trials, because for too often, we will see patients, or we'll see a drug that's been, available or being used off trial, and then we really don't get the benefit of that global knowledge of understanding if it works or how it works or which patient it works best in. So by testing them properly in these trials, it answers two questions. It's really important then to know, okay, well, if it doesn't work, let's stop. Let's, move on. We don't want to keep treating children with the patients. We need to fail early if we're going to fail. And then if we do see something that works, then we need to understand why and how to make it work even better.
Well, you definitely sidestepped my question, and I fully understand why. But one thing I wrote down from early on in your presentation, which I went ahead and circled on my notes here, is that to be cutting edge, you have to try things that you've never tried before. And I think that really defines what you've done there, and really that leading edge, and the whole reason why we see you as an innovative researcher and one of a kind. So we appreciate you joining us here, Dr. Zeigler. And I also wanted to share with the group here, just as some parting notes on it, is that you can find out more information about some of these trials. You can find out information also about, for example, the Target trial that's going through with Connect, and also some updates on, particularly if you're interested in the AMXT DFMO update. All of this can be found simply at DIPG.org. Matter of fact, on here, this is the official conduit on how Ammonex, the company that is helping to support that trial, is providing information out to parents out there. You can simply go up here, search bar, type AMXT, and it'll pull it up pretty quickly. As well as finding information about the Target trial through Connect as well. So thank you very much, Dr. Zeigler, for joining us. Thank you, everybody, for being part of this session. And we look forward to hopefully having many of these for you as we go, in addition to what's coming up at the symposium. Thank you very much.
Thank you.